Hello, I am Carl Gelhaus, Principal Scientist at MRIGlobal. With my colleague Luca Popescu, we will discuss COVID-19. So we would like to talk about COVID-19 vaccines and therapeutics. And I'd like to begin the discussion with an overview of how to make a drug. So you can see here, there's a long pipeline, a long path to a drug being approved for use. At the bottom of the screen, you can see that it takes a lot of time and a lot of money. And at the beginning, you start out with a lot of different drugs. And each of the gray circles represents a drug that will ultimately fail. And each of the black circles represents a drug that will ultimately succeed and move to the next phase. So you can see in the beginning, you do sort of discovery testing where you look for low-cost, high-throughput assays where the drugs, many drugs, maybe even 100,000 drugs will be screened and then moved forward. And those drugs that are then identified that can work will then move on to preclinical development where they'll be examined for safety and toxicology in animal models, as well as some preliminary efficacy data in animal models to show that there's a likelihood of the drug working in humans. At that point, you will go to the FDA and ask for investigational new drug status, and then move to human clinical trials that occur in 3 phases. In phase 1, it's largely aimed at efficacy, or largely aimed at safety. Phase 2 is largely a mix of safety and efficacy. And then phase 3 is a large-scale study, mostly looking at efficacy, but in all human clinical trials, safety is always important. Once you've acquired data that your drug is both safe and effective, you can apply for a new drug application or a biological license application, depending on the nature of your compound. And then you can move into licensure. Now, in the case of COVID-19, we are looking for ways to go faster. Obviously, a 10 to 15 year timeline for drugs or vaccines is not going to work. So some of the things we're doing here at MRIGlobal is we are in that discovery and in vitro screening phase. A lot of people are bringing forward products that already have been used and are known to be safe in humans. And so we would take those drugs and look at our in vitro assay. This is the virus neutralization assay. We've tested well over 150 therapeutics. At least 25 different clients have brought us candidates at this point. And what we do is we take vero cells. These are a cell line that came from a monkey kidney cell, or monkey kidney, and we expand those onto a plate. And if you look at this middle panel here that says no CPE, that's healthy vero cells that have not had any cytopathic effects from the virus. These cells are actually uninfected. Next to that, you see the panel that says CPE. That's where the virus has introduced the cytopathic effects. And as you can see, the virus has gotten small, or the cells have gotten small and granular and rounded up. And this is what it looks like when a cell has been infected with the SARS coronavirus 2. What we can then do is introduce into these cultures a drug at various concentrations. And that will show us if that drug can stop the virus from turning these healthy cells, like in the middle panel, into these diseased cells on the right panel. And if that drug can do that, then we can advance that to the next stage. If you look here on the lower left, that's sort of the format. We do this on a 96-well plate, so you can get multiple drugs, 3-4 drugs on there, multiple concentrations, 7-8 concentrations. And we can get a sense of how much drug do you need. We can also make other adjustments to the assay. Can you incubate this with the virus, incubate your drug with the virus? And does that inactivate the virus? Can you incubate it with the cells prior to the virus? And does that generate some protection within the cell itself that stops the virus from invading the cell? We have lots of different approaches to be able to do that. This is what sort of the outcome looks like. So this is, of course, we do this testing in our BSL-3 lab wearing our suits. to make sure that we don't get infected with the virus, and handling it in all these different assays. And so we get this sort of readout here. This is one example of a drug that we tested, this one back in April. And you can see that 10 and 3 micrograms per milliliter of this drug was protective. The first drug was protective, and then a couple of the others were protective, a little more low, maybe even 1 microgram per mil. We take those data points. So each of those data points represents a well on a 96 well plate. A negative means no cytopathic effects. A positive means cytopathic effects. And then we enter that data into a curve fitting software program to find out what the median effective concentration, or the concentration with 50% of the cells are protected from the virus with that particular drug. And so we calculate that out. In this case, the drugs came out to about 1.7 micrograms per milliliter was the effective concentration, which is definitely in a druggable range. There's lots of compounds that are being tested at this point. So as I said, we're testing hundreds. I wanted to give some sense of where we are, in the country as far as testing. Hydroxychloroquine has come up. MRIGlobal has tested that compound, and we didn't get the best results. And so it did have marginal efficacy, but it also had high toxicity. And so we don't really use that one here. Remdesivir, which is made by Gilead, we have used it. And we do continue to use it in our assays as a positive control. It does appear to be fairly effective. There's a lot of different people doing a lot of different things in this space, but I went with one of the high-profile funders for this is the Biomedical Advanced Research and Development Authority, or BARDA. They are certainly putting large dollar investments into COVID-19 therapeutics. And here I've listed several of the compounds that are being developed, and you can see multi-million-dollar efforts to bring these treatments forward for protection against COVID-19. And that really sums up everything I wanted to say about the therapeutics portion. I'd now like to introduce my colleague, Dr. Luca Popescu.
Hello, everybody. My name is Dr. Luca Popescu. I am a DVM with a PhD in virology. So I've been heavily involved with all the COVID research here at MRIGlobal alongside Carl. And I've been working also on some animal model development. So I'll talk a little bit about that, and then I'll touch on vaccine development also. So with vaccine development, this is kind of just a pretty high level overview, but obviously the first thing you would want to do is pick a target. This is a portion of the virus that's known as the antigen that the immune system will target to create antibodies. For SARS-CoV-2, the preferred antigen is spike protein, and this is because it is antigenic, so it does, the virus produces a lot of this protein. It's immunogenic, so the immune system of the host likes to target it. It's available to T and B cells for them to pick up, and it's also involved in receptor binding. So it's highly conserved since it has to continuously interact with the host receptors. The next thing to do is to pick a platform or an adjuvant, or pick a platform and an adjuvant, I should say. So the adjuvant is something that will trigger the innate immune response, stimulating both cellular or T cells and humoral or antibodies. And there's a few different ones. Messenger RNA has been widely used in the effort against COVID-19. So the antigen is encoded as an mRNA to be expressed by the cells. There's also different viral vectors that are replication deficient. That are used to encode the different target sequences from the virus of interest. A lot of times it's adenovirus. Some of the other lead candidates are also using vesicular stomatitis virus. And there's also a lot of proprietary adjuvants that elicit an innate immune response. There's quite a lot of candidates, and that's a good strategy to have. You want to have a lot of candidates because you want to play around with different combinations of these different components, different adjuvants, different vectors, different antigen sequences that you can kind of plug and play and see which one works best and creates the best immune response and the best protection. So if you have some good candidates, the next thing to do is to demonstrate safety and immunogenicity. So typically you would start with mice. In the case of COVID-19, we're actually starting with hamsters. That's the preferred animal model. So you want to measure the antibody levels produced and measure that via virus neutralization assay like Carl talked about. You also want to measure and characterize the T cell and innate immune response, antigen-presenting cells, cytokine pathways that are activated, and typically this is done with flow cytometry and ELISpot to test the T cell populations. Kind of the old school approach focused more on antibodies than on the cellular immune response, but more recently, I mean, within the last 20 years, I should say, it's become apparently obvious that the cellular and the innate immune response play a big role in the efficacy of vaccines. There could be some, adverse reactions. These could be minimal, like an injection site reaction with some swelling and redness. Low-grade or transient fever is still considered pretty mild. But you do want to do a full necropsy on your study animals with histopathology to confirm that none of the other organ systems are affected by your vaccine. You don't want to do more harm than good. And then in the phase 1 clinical trial, you would demonstrate safety and immunogenicity in people. Typically, these are done after the animal efficacy trials. But in the case of COVID-19, and kind of like what Carl was talking about, since we're trying to accelerate the development of vaccines and rolling them out to the general population, these have kind of been done simultaneously in these efforts just to speed up the process and make sure that we can get the vaccine out as fast as possible. Next, you also want to show efficacy. So like I said, you have a small animal. In the case of COVID-19, it's a hamster, and then also a large animal model. And this is still kind of being developed. The rhesus macaque is probably the closest thing in terms of like genetic relatedness to humans that does develop a COVID-like disease state when infected with the virus. And these all have to be done in a BSL-3 environment or an animal BSL-3 environment. And then after you show efficacy in your animal models, you progress to a phase 2 and 3 clinical trials in people. You also want to have a manufacturing and scale-up strategy. This is especially important with COVID since it's such a widespread global problem. We'll need billions of doses, and the sooner the better, obviously. So just to elaborate a little bit on the small animal model, is the Syrian golden hamster. We have done some work with this ourselves, but these are important for efficacy studies. They are infected intranasally, and that kind of replicates the natural route of exposure in people, since this is a respiratory disease. The ACE2 receptor is very similar to humans and hamsters, and SARS-2 appears to be very promiscuous. People have been speculating that it jumped from like bats to humans, so it likes to play around with different receptors and is available to infect different receptors. This hamster is good also because it does show clinical signs. It has labored breathing, it has pretty severe weight loss, it shows lethargy, but then it does start to recover after around 7 days. And this is pretty typical for healthy humans also. And these animals, most research animals are young, healthy animals. So it does mimic that disease, but there is a limitation since, we tend to use younger and healthier animals. And then to measure the disease progression, you would want to do lung pathology, histopathology, and look at the lesions that develop, in your animal model. And then also look at viral levels in the lungs, the trachea, nasal turbinates, those are the channels in your nose, salivary glands. And there's also been definitely been cases in humans showing that SARS-CoV-2 causes a GI disease as well. And we have, and others as well have noted that, the virus can replicate in the intestines. The large animal model, like I said, is the rhesus macaque. That's the one that's kind of the favored one so far, because these non-human primates are close to humans, and because they're bigger, you can have some more refined disease markers. You can look at cytokines, cellular responses, and viral replication dynamics. You have more samples to work with, and kind of can characterize the disease a little bit better. Like in people, older rhesus macaques develop worse disease, and it's a similar respiratory syndrome as in people. You have labored breathing, rapid breathing, weight loss. There have been reports of transient fever too, although that is rarer than in people. And you do have to use a pretty high viral challenge dose, and you have to inoculate simultaneously via a variety of routes, including intratracheal and intranasal. And then again, you want to measure disease progression. Because these animals are so precious, we tend to avoid sacrificing them at all costs if we can. So we have more refined techniques for assessing disease states. Initially, people did a lot of X-rays and CTs, and X-rays can show some degree of pneumonia in the lungs. But CT is a lot more sensitive, and even then, the changes are usually very transient. They're only there for a couple of days. So you will do necropsy at the end of these studies, just to assess the level of damage and recovery of the lungs. And again, then you want to look at viral levels in lungs, trachea, nasal turbinates, salivary glands, and intestines. And like I said, we do have more refined measures for these as well. You don't have to sacrifice the animals for this. You can do viral levels in lungs via bronchoalveolar lavage. You can do nasal washes or swabs, and also swabs in the back of the throat to assess viral levels that way, which is, again, closer to humans, what you would do in humans. So then to go over kind of the big players in the vaccine realm that have come up. Obviously, it's gotten a lot of hype. They had previously been working on a SARS-1 vaccine, so they kind of had a leg up on a lot of people. But there's quite a few vaccine candidates that have made it to phase 3 clinical trials. Like I said, Moderna, Pfizer is another one, AstraZeneca, that are collaborating with Oxford. That's another big one. And then Janssen also has a vaccine that has made it all the way to phase 3 clinical trials. And then Novavax and Sanofi are in phase 1 or 2 clinical trials. And at least according to BARDA, Merck was not in clinical trials as of today. Everyone is kind of targeting the spike protein. That's the preferred antigen that everyone's using. So, like I said, there's a good reasons for targeting spike, but there's also some limitations to that. So I'll go over that a little bit now. The vaccines themselves can elicit a very strong immune response, and that can actually worsen the disease, especially with, we've seen with COVID-19, there's been lots of cases that it causes this kind of cytokine storm. And with previous vaccines developed to SARS-1, that was kind of a problem that you saw, these strong cytokine responses, and antibody-mediated enhancement also is seen with a lot of different viruses, including, influenza, West Nile, yellow fever. So this is why efficacy studies in animals are important. You want to weed out these adverse effects and any bad things that can happen in that way. If you elicit too strong of a response and it causes worse disease. Breadth of protection is another big one. That spike protein, because it interacts with so many targets and because the virus produces so much of it, has been shown to mutate quite readily, and it actually mutates at the furin cleavage site, which is an important host. Furin is an important host enzyme that facilitates the virus kind of replicating and entering and doing things in the host cells. So how much is the virus mutating? We obviously know that the US isolates are different from Italian or French or Chinese isolates. So will the vaccine that we generate protect people all over the world, or does it have to be a vaccine for each of these strains for every, part of the world, essentially? And then will the virus actually escape the immune response, the natural immune response, or the vaccine immune response, which is essentially what influenza does every year, right? This is why you have to get a flu shot every year. And certainly there's been reports of people getting reinfected with COVID-19. You get infected, it's not that bad, and then you can get it again. And in some cases, it can actually be worse. It causes a worse disease. So there's still a lot of unknowns about that. Obviously, epidemiology and virus tracking are very important, but it takes a lot of time to, amass a lot of data to say that, something is significant or insignificant. So whether the spike protein is the best candidate is up for debate, and I'm sure it will be up for debate in the coming years as more vaccines and therapeutics get developed. I think certainly it's a good place to start. And all these limitations are definitely not throwing in the towel or anything like that, but it is highlighting that viruses evolve very quickly. They evolve from the moment that they enter the host to the moment that you shed your last virion. Those two things can be very different. The genetics change, and the virus that you get is very different than the virus that you might pass on to somebody else. And this has a lot of implications for vaccine and therapeutic development. And then, typically, certainly with influenza, we develop vaccines, and they tend to be more efficient at protecting healthy people, which doesn't really help the target demographic, right? With influenza and with COVID, people with immunocompromised conditions or pre-existing conditions, the elderly and the young in influenza's case, tend to be more affected by these viruses. So those populations might require a different vaccine, something that elicits kind of a different immune response that protects those people a little bit differently than they might protect someone who's healthy. So the first vaccines that get developed may not be the best vaccines, and it will have to be a continuous work in progress to develop more vaccines and therapeutics that kind of evolve with the virus and continue to address the problem that the virus poses, since it's so widespread and it's definitely not going anywhere. And certainly epidemiology and public health are important. That virus tracking that I was talking about is critical in knowing which populations are at risk, which populations need to get the vaccine first, what pockets, geographical areas have higher outbreak rates or anything like that, so we can focus our responses in a smart and efficient way. And then obviously, the thing that we can all do to curb the spread and reduce the impact, just wash your hands, try to limit your interactions with people, and just educate yourself. The science is changing, but a lot of these basic things are staying the same. So we just have to do our part, and we'll get through it together.